Hello everyone. InformationBoxTicket Lifestyles brings you today food microbiology topic on botulism food poisoning. But before watching this video, don't forget to like this video, subscribe to my channel and press the bell icon button. Let's see the table of content. First, we will dive into what is botulism food poisoning, the source of Clostridium botulinum, contamination, Clostridium botulinum neurotoxin production, clinical manifestation of botulinum food poisoning, epidemiology of botulinum food poisoning, botulinum toxin action, detection methods of botulinum food poisoning, treatments, and lastly, control measures. Let's start with the introduction of botulism food poisoning. Botulism is a potentially fatal foodborne illness that causes neuroparalysis when botulinum neurotoxins are consumed. Botulism is a rare disease with a high mortality rate. Even a tiny quantity of poison may kill millions of people. Clostridium botulinum produces intoxication, which happens as a result of either eating preformed botulinum neurotoxins in foods or producing botulinum neurotoxins in the gut. Botulinum toxins type A to type T are responsible for sickness in humans and animals. Foodborne, newborn, and wound botulism are the clinical epidemiological manifestation of botulism. Botulinum toxins A, B, and E have been linked to foodborne botulism in humans. Botulinum neurotoxins have a fatal dosage of 0.2 gram to 2 gram per kg body weight for humans. Botulinum toxins is a potentially harmful biological substance in the environment. So what is the source of Clostridium botulinum contamination? Clostridium botulinum is an anaerobic spore-forming gram-positive bacteria whose spores are widely disseminated throughout the environment, including soil, sewage, muck, lakes, sea, and ocean sediments and the intestines of land and aquatic animals. Because of the bacterium's ability to spread far, its spores can infect food where it colonizes and create toxins. Clostridium botulinum spores in honey and syrup, the primary cause of newborn botulism. Because the spores can resist high heating temperatures during processing, poorly treated canned foods are also a major source of infection. Other food sources include smoked, salt-dried, and fermented meat items, as well as numerous varieties of lake and sea fish. Type A and Type B botulism neurotoxins are also often found in soil and biofertilizers, as they are likely to infect crops and fruits. Beans, baked potatoes, corn, celery, mushrooms, onions, and olives that have been improperly cooked. Next comes the Clostridium botulinum neurotoxin production. Botulinum neurotoxins are extracellular proteins weighing roughly 150 kilodaltons generated by Clostridium botulinum. Botulinum neurotoxins have 7 unique serotypes based on bacterial physiology and phenology, ranging from type A to H. Based on their proteolytic and non-proteolytic action, the 7 toxins are divided into 4 groups: Group 1, 2, 3, and 4. Group 1 proteolytic type A, B, and F can proliferate at temperatures ranging from 10 to 20 degrees Celsius, whereas Group 2 non-proteolytic type B, E, and F may proliferate at 2.5 to 3 degrees Celsius. Type C and D are non-proteolytic in Group 3, whereas Type G is in Group 4. Clostridium botulinum is a stringent anaerobe that can withstand high temperatures. It produces toxins in the gut and causes intoxication when swallowed. In this figure, you can see in number A, molecular processes of natural C2 toxin poisoning. To bind to C2-1, heavy chain C2 type 2 must be activated by protease and oligomerized. After receptor-mediated endocytosis, Acidification of early endosomes lead C2 type 1 to be delivered into the target cell cytosol with the help of the pores produced by C2 type 2A oligomers to begin its enzymatic activity. In the number B, neurodelivery model based on the C2 type 2C1 and C2 1T transport systems. C2 type 1T is changed separately to contain a conjugated molecule as a payload for delivery through activated C2 type 2C1. This modular system has a potential to reach targets in the cytosol of the peripheral nerves. The requirements of conjugation and subsequent translocation by C2 type 2C1 restrict payloads. Kindly show your support by subscribing to my channel. Clinical manifestation of botulism food poisoning. Within 18 to 38 hours of consumption, symptoms that impact the gastrointestinal tract becomes noticeable. Abdominal discomfort, nausea, vomiting, diarrhea, and constipation are common symptoms. Neurological symptoms such as flaccid paralysis, symmetric descending weakness, slurred speech, trouble swallowing, dizziness, impaired vision, dry mouth, and extraocular muscular weakness emerge after intoxication. Babies under 1 year of age are especially affected by baby botulism due to a poorly established gut microbiota where the toxins are generated in the intestine. Infants may exhibit signs such as weak, harsh scream, ptosis, poor suck, inability to hold the head erect, and weak muscles. In case of wound botulism, the organism colonizes the infected site such as wound, abscess, or pus where Clostridium spores germinate and create toxins. Wound botulism symptoms include hypotension, breathing issues, eye muscle paralysis, ataxia, and dilated fixed pupils. In the event of fatality, that generally occurs between 3 to 6 days following intoxication. Epidemiology of botulism food poisoning. Botulinum toxin was utilized as a biological agent by the British, American, and Japanese militaries during World War II. The largest epidemic to date happened in Egypt in 1991, caused by eating of type 3 toxin found in a typically salted fish dish known as fisac, which resulted in 91 instances of an outbreak and 18 deaths. The fish was salted and preserved in barrels, which provided an ideal habitat for botulinum toxin development. In 2004, an osteopathic physician sought to use botulinum toxin on himself and his girlfriend as a Botox. Both were later hospitalized for respiratory failure and required ventilation for several months. Approximately 90% of botulinum infections occur in the United States, with 9 to 10 outbreaks reported each year. Home preserved food items, especially canning, bottling, and preserving food in oil, were linked to those outbreaks. Now let's head to the botulinum toxin action. Orally administered botulinum toxin or toxin manufactured in situ must withstand stomach acid in order to transit into the gut, where it is absorbed and colonizes in the duodenum and jejunum. The poison then enters the circulation and is taken up by endocytosis before reaching the peripheral nerve system. Once within the neuron, its chain is broken, allowing it to function as a zinc-dependent protease and assault SNAR proteins. It binds at the muscle junction and blocks the release of acetylcholine, causing the transmission of impulses to halt and resulting in flaccid paralysis. Kindly support this channel by subscribing. Detection methods of botulism food poisoning. Number 1. Bioassay. The most sensitive and extensively used approach for assessing toxin toxicity is the bioassay using a mouse. Symptoms appear in mice 4 hours after treatment. The botulinum toxin is characterized by abdominal vibration, wasp-shaped belly, limb paralysis, and trouble breathing. Number 2. Immunoassay. The most sensitive immunoassays used to determine the botulinum toxin is ELISA. ELISA binds the antigen toxin to the antibody antitoxins present in the solid surface. Then, another enzyme labeled antibody binds to the antigen, and the amount of toxins can be determined by enzymatic reaction. The specificity and sensitivity of ELISA are determined by the quality of antiserum used. Number 3. Endopeptidase assay. Endopeptidase test is an in vitro assay used to assess the toxin's therapeutic potential. Example: Botox, Dysport, Xeomin. A bioluminescent signal, fluorescence, resonance energy transfer, and mass spectrometry are used to detect it. Endopeptidase tests are more reliable than immunoassays because they only detect active botulinum toxins. Number 4. PCR-based assays. On agarose gel electrophoresis, a standard multiplex PCR identifies the particular toxin gene using a high annealing temperature primer. It is capable of detecting toxins of type A, B, E, and F. Botulinum, neurotoxins A, B, and E are detected using real-time PCR. Within 1 to 2 hours, fluorescence-based PCR amplifies gene fragments and detects the toxin with excellent sensitivity. Next is treatments of botulism food poisoning. An individual with identical botulism symptoms should be admitted to critical care and given parenteral nourishment. If a toxin is detected, antitoxin should be administered within 24 hours for maximum efficiency, since it neutralizes all free toxins in the bloodstream before they reach the neuromuscular junction. Bivalent or trivalent equine immunoglobulins are currently accessible as antitoxins. In the instance of wound botulism, medicines such as penicillin should be used to treat the wound. Lastly, control measures of botulism food poisoning. Food should be properly heated above 121 degrees Celsius and refrigerated below 4 degrees Celsius. Reduced spores development is a benefit of acidifying chemical like citric acid in canned foods. Avoid food that you believe has been improperly reheated or undercooked. And that's it. Thank you so much for watching till the end. Don't forget to support this channel by liking this video, subscribing to my channel, and pressing the bell icon button so you don't miss any of my videos. Thank you.